introduction. People are starting to roll in now. Okay, so welcome everyone to the October Myeloid Network seminar series. I'm delighted to introduce Dr. Max Krummel. He holds the Robert Smith Endowed Chair in Experimental Pathology and serves as a professor in the Department of Pathology at the University of California, San Francisco. He has more than 15 years of experience in developing groundbreaking research in immunology and has made significant contributions to understanding T-cell responses and immune regulation. I think very significantly and importantly, it's critical to mention that his early work involved the development of monoclonal antibodies targeting co-stimulatory and inhibitory molecules on T-cells, notably contributing to the creation of the cancer immunotherapy drug that we all know, which is ipilimumab, and was the first FDA-approved therapy for melanoma. Max's research continues to pioneer the study of immune system dynamics using advanced imaging techniques to uncover new pathways to modulate immune responses in the tumor microenvironment. And so his current focus is on the spatio-temporal dynamics of immune cell interactions, with a particular emphasis of how T cells interact with other cells, such as dendritic cells and macrophages and complex tissues. And he utilizes cutting-edge imaging and molecular genetics to reveal novel insights into how the immune system coordinates responses to disease, with the goal of improving therapeutics for cancer and other human disease. It was his group that was the first to perform live imaging events in progressive tumors, in which incoming tumor-specific T cells are captured by a population of myeloid cells. And so his work on live imaging of tumor immune interactions has led to new approaches for protein immunotherapeutics and continues to shape our understanding of how the immune system operates, both in health and disease. And so, Max, we're delighted to have you here. We're excited for your talk. I also want to thank our sponsors, AstraZeneca and Charisma Therapeutics. Also thank the University of California, San Diego, who supports this program. And another reminder that the Myeloid Network is partnered with SITC next month to run a co-sponsored session during the annual SITC meeting. So please join us there. There will not be a Myeloid Network seminar for the month of November since we're hosting the event at SITC. So with that, Max, I'll turn it over to you, and thank you again.
Great, thanks for having me. Let me see if I can get a screen shared here. I think you can see this.
That looks great.
Super, all right. Let me get my screen slightly organized here. Well, thanks everyone for coming. Hold on one second. What's going on? I'm going to stop sharing and just check this out. Looks like this thing wants to advance. Okay. Once again. Okay, thanks for having me. And here we go. So there still seems something a little bit off. I'm sorry. Give us a sec here. So I want to talk about the concept of emergent behaviors in immune systems as a kind of an entry point for talking into the myeloid space. And the idea here is that if you think about how molecules are configured, the emergent behavior of a collection of molecules being configured is essentially gets you to the stage where you can think up a level to the subcellular. So the subcellular, obviously the things that organize how individual cells behave. And I want to play you a movie as a kind of a spatial and temporal concept to orient what I want to talk about today. So this is a movie that shows, it's from lattice light sheet imaging, which is a way of taking very thin planes at around 200 nanometer isotropic resolution, so that you can see things like microvilli and the veils on things like dendritic cells. So this little ear right here, if you look at it in a normal microscope, if you got it on edge, it would look like a dendrite, but it's more of a veil. And these little, for example, these little puncta on the T cells are the microvilli that T cells use to scan. So as this movie plays, you're going to get a sort of three-dimensional view of, these are mouse T cells and mouse dendritic cells, by the way, of the T cell, how it scans. So it uses these finger-like projections to feel out the world around it. And you can see how it interdigitates to try to push into the neighboring cell. If you take a professional antigen presenting cell like a dendritic cell, though, you see this other really cool behavior, these cutaways where the dendritic cell essentially forms the puzzle piece opposite to the T cell. It doesn't form microvilli, but instead it forms these kind of converse pieces that slip into the border. And what it essentially is allowing the T cell to do is to survey using the entire tips of all the membrane what it finds there. And I use this as an example of a concept of partnerships in the immune system to remind you that all the cells in our body are individual, and yet they have gene expression that is partnered so that they can do the function they need to do. T cells need to survey, and dendritic cells want to present. And so the gene expression is actually partnered to make that a very efficient event. And I like that movie for the example of it. So that's an emergent behavior that's a consequence of all the molecules in the cells that are expressed, including the functions of various actins and ARP2/3s and dynamins, et cetera. If you go up a level from that, the way that cells behave gives rise to the way that tissues and organs and tumors behave. So you think about the multicellular sort of level, and that's where we're going to focus a lot of effort today. The final emergent behavior, which is the one that largely fuels a lot of the interest in the work that all of us do, is the idea that the patient experience, the clinical, here's time versus outcome. First with IPI or versus IPI Nivo, two checkpoint drugs. So that's ultimately a function of how the tumor microenvironment, the multicellular, is behaving. So I also want to take a moment to remind ourselves that although cancer immunology is one of the things that has been very much orienting towards immunology in the last 20 years, really, sorry, I've got somebody in the way here. The immune system has been understood to be something different than what we thought about maybe 20 years ago. And that is to say that immunology maybe was defined by the concept of response to pathogens, and with cancer maybe being a extension of that. But I very much think that the work that's been done across brain, heart, pancreas, infectious disease, respiratory diseases, et cetera, have pointed us to the idea that the immune system really has a series of modes or roles to play that it can assemble in different formats, not just to get rid of things that you want to get rid of, but also to tune the things you do have. So for example, right now, you won't remember this talk if it weren't for the microglia, the macrophages in your brain that are pruning your synapses. So that's not a defense function. That's a making you better function. A similar example would be that T cells and macrophages and myeloid cells in your gut are essentially forming a quarantine or even a curation of your microbiota. You have T cell receptors that recognize the products, the peptides of some of your key commensals, and they can essentially titrate out the amount of microbiota you have, again, kind of making you better. Immune system is also coming into all these other settings where we recognize that the modules, for example, we know that macrophages are critical for adipose and for lipid deposition. So those can go awry. Now we understand, for example, arteriosclerosis, which used to be considered a disease of lipid metabolism and is now equally well, I think, understood to be a disease where underneath all arteriosclerotic plaques, you have angry macrophages. So I'm going to refer to this concept as archetypes in this talk. And really the idea here is that on the left-hand side, if you see immunity as having this kind of classical spectrum that goes back to famous pioneers in immunology, like McFarland Burnett, for example, posited the idea of clonal tolerance, that your immune system could be sort of inactive in one way or against things versus destructive. And that was how you defined sort of self at one end and non-self at the other end. That there's a huge area that's opening up in the last 20 years that represents these, I call them sort of accommodation archetypes. Namely, this is here the archetype is defined as collections of cells in linked states across tissue types. And I'm saying typically following an evolutionary design that might be involved, for example, in using the immune system to assist tissue development. So for example, Zeno Werb some years ago showed that macrophages and T cells were important for breast duct branching postpartum. We also know, for example, that neutrophils and macrophages are important in healing wounds. So some of the same cell types like macrophages can be involved in both, but the nature of the macrophages and the gene modules that are expressed in them can be variable, and they end up like that first movie I showed, forming partnerships that essentially does what's shown in these hexagons. Tissue development, tissue metabolism involves Tregs and macrophages. So I'm going to introduce this idea, and this is why I talked about emergent behavior of a higher level of abstraction than a single cell type, but not as high all the way as like health and disease. It's sort of a recurrent motif. And this represents also highly specific sensing populations. So there's frequently T cells involved that are sensing, even actually now in the healing of wounds. Some people may have seen Yasmin Balkade's work, where T cells have long been known in rag mice. Skin wound healing takes longer. So you have highly specific sensing populations like T cells that can be partnered with pattern recognizing cells like our favorite macrophages. So when we think about tumor immunity, I think it's fair to say that we really want forms of sort of archetypal states of the immune system that are configured to have sort of optimal killing activity in one way or another. So the classic collection of cells that I'll describe in a moment is called CD8 T cells, CDC1s, and NK cells. And we first isolated CDC1s as being a critical player in these modules back in 2014. I think it's now become even more popular, this concept, and more evidence is building for this. I'll show you, we've known for some years. There's also reactive archetypes that involve CD4 cells and stimulatory CDC2s that are also functional in cancer. So it's fair to say that we can think about the idea of reactivity of an immune system that brings together collections of cells to make sort of an optimal behavior. And then these forms that I think I'm going to show you are likely the format for which cancers evade immune responses, which I'm going to call the dominant immune responses in tumors. So the immune system has been harnessed by tumors, and it's been harnessed in fairly specific ways depending on the tumor. And this just brings up the idea that within a tumor microenvironment at start, you may be very much biased towards these red dominant formats. The immune system may be kind of configured in that direction with just a sliver, maybe sometimes of reactive immunity, sometimes almost none. This is on a fulcrum. And then a successful therapy, in some ways, immunotherapy, is defined by the fact that at the end of it all, you have a very strong reactive system with lots of T cells poised to do killing, for example, and relatively little of these dominant responses remaining. So this just summarizes some work that we've done over the last really 15 years or so. Again, this particular one is being picked up very much now in the hub concept. And that kind of comes down to the idea that we isolated that CDC1s were really essential for CD8 immunity in tumors, and that they are actually fed by lymphocytes, including NK cells and sometimes T cells themselves, to make this module sort of effective. Those dendritic cells can traffic to the lymph node where they can activate CD8 T cells. And then the CD8 T cells can come back and meet those CDC1s again in the tumor microenvironment, leading to elimination. So that's kind of a general, for us, an archetype. It doesn't have to be a hub. It doesn't have to be spatial. There are components of their hubs, but then there's also components of this that can be distributed. And then I think, in contrast, or at least what seems to be a sort of a separate module is the ability for CDC2s, for example, to do something similar, go to the lymph nodes and trigger CD4 cells, and then the CD4 cells can come and be reactivated there on the same CDC2s, that very much being regulated by regulatory T cells. These are kind of two ways to view reactive immunity that we want. But what I want to talk about, and this will dive into some myeloid diversity as we go, is what is or are the dominant immune systems in cancer? And in some ways, one way of asking that is, what does the immune system think it's doing there? What has it been programmed to do? Which is to say also, how do we even classify such higher order biology? Again, the reason I talked about emergent behaviors is that different emergent behaviors can come from different collections of molecules leading to cellular behavior, leading to those higher order biologies. And for us, it's also, how do we try to modulate that higher order biology? So if you don't know what you have, you don't know what signals to give. And so we've been working towards that. The way we did this was this is a study that came out just 2 years ago, but continues to evolve and expand, in fact, in which we took tumors from about a dozen garden variety cancers, and we got all comers with the whole goal of seeing sort of a diversity of tumor microenvironments. We didn't want to just stick to one indication. And we took biopsies from those, and we did high dimensional flow cytometry, and we also did high dimensional and relatively low dimensional RNA sequencing. So back then, the original thing was to sort T cells and regulatory T cells into individual bins and look at their RNA-seq, so what are they genes are they expressing? So one of the things you can imagine you can do from this, for example, is in flow cytometry, as an example, looking at T cells, you can go across indication where this spans from hepatocellular, head and neck, kidney being very high in T cells to like GBM being very low in T cells. And you can waterfall and you can say, okay, so some tumors are more T cell rich and other ones are more T cell poor. And you can do the same thing for myeloid cells. And some of the ones that were high, like kidney and T cells, are also high for myeloid cells. But other ones like head and neck that are high for T cells are actually relatively low on average for myeloid cells. And you can do the same thing, for example, for stromal cells. So this gives you a kind of a landscape, or like what people might call an atlas. But I think what we're going to move towards is the idea that what it allows you to start to think about is what are some of the combinations of these archetypes of the immune system that can be built. So if you look at myeloid cells, I think you'll get a flavor of what I'm going to show you, which is to say that in PDAC, pancreatic ductal adenocarcinoma, really there seems to be sort of two classes of tumors that you can find in people, those that are really high in myeloid and those that are relatively low. And this really shouldn't be a box and whiskers plot by all statistical virtues. And as soon as you see that, you realize that that same trend exists. The spread of distribution is such that if you picked a given tumor from somewhere in here, you wouldn't know what tumor type you're in, even though there is a general trend. And this brings up the idea of, again, about how are these all things combined? If you're not always T cell high, myeloid low, for example, then how, in fact, how is this all organized? So we did a lot of different kinds of clustering of this data, from small, very low dimensional to high dimensional frequency data here, to gene expression. And the sort of the best so far method actually just involves 10 features of a tumor to say, like, for example, if you were a cytotoxic tumor, you would expect to have lots of cytotoxic T cells. So the frequency of a cell in a tumor indicates something about the overall archetype or biology of that tumor. And so we're using 10 different frequencies, how many T cells you have, how many myeloid cells, how rich are you in stroma, and then within the T cell compartment, are you rich in particular subsets, 4s, 8s, or Tregs, and within the myeloid compartment, what are you rich in? And that leads to the ability to make yourself a UMAP. And similar to the same ones you see for single cells, now every dot, though, is a patient. And if you do this, you see, for example, that you can cluster based on, here's given some sort of conceptual names. These red in the top far left are immune rich, and they have lots of CD8 cells and lots of macrophages. These ones are somewhat similar. They're also immune rich. They have lots of immune cells overall, but they have more monocytes. And it's, again, just using these 10 features to define them. So to get another way of looking at that, for example, you can see that if you just plot T cell density, this bottom left-hand side of the map is where T cells are most rich. And so you see familiar indications that we know are T cell rich in humans like melanoma and lung and kidney. And just as a comparison for future reference, the area between about 12 o'clock and 3 o'clock is where we find 3 forms of immune deserts. And although we think deserts means not a lot of immune cells, there really are variations in these deserts, and they're functionally quite important. Though they do all, of course, in a map like this that's based on density clustered together. So then if you overlay the tissue of origin to ask yourself, like, is there a trend here? There are. So for example, now in this left-hand side overall, the red dots mean kidney cancer. And about 80% of kidney cancers then live either where we had these immune-rich CD8 macrophage populations or below where we have immune-rich monocyte populations. But you can also find red dots over here in the immune desert, the 12 o'clock to 3 o'clock region. That it tends to be rich, for example, in things like gynecological tumors that we know tends to be fairly sort of, again, immune poor. You can find down here, you can find lung cancers and melanomas in blue and brown, respectively. But again, you'll find some lung and melanomas that are up here in these immune deserts, and even some over here that look a little like kidney cancer. So one of the things that I think this work is starting to emphasize is that the indication that you find can have a bias towards what kind of tumor microenvironment you have, but it's by no means prescriptive. And I think the way to think about this is that as tumors grow out and they start throwing mutations, they're looking for a combination lock to solve, and they can solve one of any of these 12. And if they do that, then they end up living. So this concept of the dominant immune archetype, it actually compartmentalizes other immunobiology, even though we didn't choose to do that. In other words, we created classes of tumors based on 10 features, um, and yet other key biology segments only into one or two of these classes. So for example, natural killer cells, you can see, are really only high in the pink, a little less so in the red archetype, and just a tiny bit in this blue T-cell centric form here. B cells, on the other hand, turn out to be very enriched in this TC-MAC phenotype that you find right here. So other biology is parsed into one or sometimes just two of these classifications. Like we didn't cluster based on mast cells, but mast cells turn out to be just in this particular clusters where we have lots of them. Plasma cells, the form of the TH gene expression, GATA3, et cetera, is also very much parsed by these archetypes. So is exhaustion biology, Treg biology, et cetera. And as I was alluding to too, that this desert region here from 12 o'clock to 3 o'clock shows you that what we're finding by looking at the fundamental collection of immune cells actually also somehow relates to the way that the tumor got there. And one of the easiest ways to show you that is to have overlaid the tumor's expression of Ki67, which is a particular way that a tumor can get through the cell cycle to keep dividing and dividing and dividing. And what you see when we overlay the heat of Ki67 gene protein expression onto the same map, you can find that exact sort of immune desert region very much is highlighted by Ki67. So there, the tumor, its biology, has a very strong relationship to what kind of immune system it's brought in. And these are things that we continue to expand on. In fact, we can also find mutational landscape, particular genes that frequently are mutated in tumors. They parse into individual archetypes as well. So, kind of to summarize or even extend this a little bit further, this concept of being able to look at the nature of the immune system that you have, its component cell types, the gene expression in those cell types, their related cells that come with them, are really important. So if you take kidney cancer, where I said about 80% of the patients are either pink or red, with the remainder now being shown here as yellow as a single class, you can really see that it's much better to be those CD8 monocytes that have lots of NK cells, for example, as opposed to the CD8 macrophages. That phenotype, the archetype there. There's also a degree to which we keep finding more and more biological consistency with these classes. So for example, now we're showing you here in a heat map just the 12 colors that represent the 12 archetypes, and down below in columns are chemokine families. We didn't organize them by family, but they self-organized that way when we clustered our patients by high, low expression of different chemokines. And you can see, for example, that the red and the pink are very high in a collection of chemokines that turn out to be the T-cell recruiting chemokines. Some of the stromal ones have their own unique signatures. And for example, the immune deserts have very little chemokines overall. So again, we didn't cluster based on chemokine frequency. We clustered based on cell frequency, but we got back, via consistency argument, we got back chemokines. They're the things that recruit the cells. And I think perhaps the most exciting part of this for me, as a just thinking kind of in scholarship way, is that when we think about the immune system then, and we look at these 12 archetypes, again, given the same color coding, if we push them up against what we know are the immune systems in other settings, you can make these very, very clean connections between the cell types that are really abundant in these first two that are prominent in kidney cancer and what we find in chronic viral infections. Some of these ones that I talked about that have, for example, B cells in them really look a lot like the these are plasma cells, it turns out, extrafollicular responses. These resting B cells in that one that I showed you are really look a lot like actually quiescent lymph nodes, which surprisingly, but actually not surprisingly, quiescent lymph nodes are tolerant. They're not active, and so a tumor would do well to look like that. Fat metabolism seems to be a really likely model for these black archetypes. And then, as I said, there's these 3 immune deserts, and they really vary. 2 of them have connections to various stages of skin wound repair, and possibly other wound repair. And another one really doesn't. It really actually looks a lot more like what you see in neuronal inflammation, something I'll get to in a moment. One of the reasons why we think this idea is important is shown here. This was a patient that came to us in just this last year, a patient really to a very good friend of mine, and had a sarcoma. And typically, you might say you have a sarcoma, you treat it like all other sarcomas. Of course, sarcomas are one of the more diverse tumor types, but in general, if one were to map sarcomas in general, and just say if you're a sarcoma, they tend to be these immune desert CD8s. So if you look in here by tumor origin, a lot of pink, which means sarcoma in the tumor origin map. But within a day of getting the tumor microenvironment from the sample, we knew that this patient lived actually more in this immune desert CD4. And what's interesting about that, as I mentioned this moment ago, it resembles, it's an archetype that resembles neuronal inflammation. It also has as its dominance populations, for example, of tumors being GBM and things like some of the GYN tumors, the MSS GYNs. And so these are immune deserts. Just to kind of go back to a review that we wrote about this concept of archetypes, is that when we look at the, what defines immune desert CD4 macrophage populations, it's a lot of CAFs, decent amount of neutrophils, really defined by this axis of chemokines, for example, CXR2, a lot of nervous system gene expression. And it kind of looks like it might be tumors that have adopted a pattern of neuronal inflammation that at some point is presumably quite inhibitory to T cell activation. And in fact, by the time we did single cell sequencing on this particular patient, we knew that we validated, in fact, that they had high expression of this axis, the CXCR2. They had low Treg but high Th17, another thing that we had seen here. And their top mutated genes were all neuronal. So, we're starting to see predictive power of having classes that seem to look like a new person comes in and you can actually say, well, this is the type of biology you have. And I think you can easily kind of extrapolate how that's going to lead to the point where we can treat patients by their archetype rather than by the name of their disease. We probably shouldn't treat this patient as if they were an IDCD8, for example. And then I want to take one other little gander for everyone's purposes down, a comparative biology, sort of evolutionary biology of immune microenvironments. And this is basically comparing 15 mouse tumor models in a huge collection of either B6 or BALB-C, and some of them are ectopic, some of them are GEM models. And we picked ones that represent somewhere on the order of 99% of all tumor immunology studies. So we tried to not have we obviously didn't want to have 100 mouse models, but we picked sort of 15 that represent your standards. And you'll see them over here as being, the ones you'll recognize: B16, ID8, 41, LLC. So if you look at the frequencies of immune cells in human tumors versus mouse tumors, you can see very easily that T cells are really quite parsimonious and sparse in mouse tumors overall compared to humans. Myeloid cells, on the other hand, tend to be much more populous in mouse cells as compared to human. And the populations are even more evidently different when you take that myeloid population and you look at macrophages. There's just a way abundance of macrophages in mouse tumors compared to in human, on average. And this average turns out to be important. The variance here shows where tumors vary in humans versus in mouse. In mouse, you have myeloids of various different densities, and that's the main source of variation. But in human, the main source of variation is actually in T cell density. And what this leads to is if you do the same sort of clustering based on composition between mouse and human, the human now are the dots, and you can see the same sort of thing, but it's actually mirror image. The red and the pink are kind of together, and here's those immune deserts that were between 12 and 3 are now between 9 and 6 or something like that. 7 and 11, whatever that is. But this is where all of the mouse tumors are. Most mouse tumors look like human desert immune responses. There's one exception, Renka, that stands out. It stands for renal cell carcinoma. It's a BALB-C model. It actually looks a decent amount like some of these renal carcinomas that live in these red and pink archetypes. But if you just basically do a cosine similarity matrix, this is just a way of representing how similar things are. You find basically all the mouse archetypes are together, and most of the humans are together, but they're not intermixed. So you don't really have a lot of mouse models that are particularly good at representing these broader distributions of cells. something to be dealt with. And in a longer talk, I could show you some of the ways that we can now deal with these. But again, I want to bring this back to the kind of the importance of thinking this way, because archetypes basically encapsulate target biology. If you were to treat with a drug that is meant to hit the interaction between CD8 cells and macrophages, for example, it's sort of useless in a lot of ways to try to do that in settings where those cell types aren't present or that cell biology is not present. So this idea of cutting the pie of tumor microenvironments and creating wedges that represent sort of a shared biological signature is to be able to think about how to target that signature and bring it across to something that's more reactive. One last thing on these before I get into a relatively recent story from RJ Ray in the lab is that we can think about archetypes also as from cell types. And what you find is that the gene expression within a myeloid cell, for example, in an archetype will vary. So this is actually a more complex UMAP, but what I've highlighted in green here are the expression of various genes that you probably recognize. For example, MRC1 is CD206, CD163, I think everyone knows, arginase. Within the myeloid compartment is Z-scored across the rows to show you that, for example, CD163 tends to be very highly expressed on macrophages. This is gene expression within the myeloid population. It tends to be fairly highly expressed in these stromal rich tumors. Arginase, for example, is quite highly expressed in one of the three immune deserts, as well as one of these is what we call the extrafollicular response looking tumors. But really, not so much across the other ones. So again, the ability to compartmentalize what kinds of cells has gotten you there is also compartmentalizing what sorts of gene expression within those cells is present. Presumably, again, that's the origin of the emergent behavior of the cell collection. So I'm going to tell you a relatively recent story. This is just accepted now at JakeSmed. It's also in bioRxiv, about how we think about building more of the reactive archetypes. And the example given here is the type 1 reactive archetypes, where we have these CDC1s. We have a role for lymphocytes in bolstering those CDC1s, and then CD8 T cells are the ultimate executioner and recipient of that. We think about that as a partnership, and the question is, how can we think about using immune building blocks to create that? And really the answer, interesting, was from a study of basic macrophage biology based on another different hypothesis. So I want to remind everybody of something that we should be clear, is that this is our work on the left, just taking apart myeloid cells in the tumor microenvironment and using statistical computational methods to cluster them. And so this is from 2022. Essentially, you see, for example, this is a PYMT tumor, but we show in the same paper, you see the same sorts of populations in B16, but also even in human, where you can find elementary monocytes, here lysis C positive. You can find your C1q, very mature TAMs, and you can find various populations that are along the trajectory from pink to dark red, with gene overlays that represent sort of, I think, modular expression, like the more ISG, the stress responsive, or as I'll show you in a moment, are also another name for hypoxic macrophages. And recently they've also been noted, although we notice it's too, they're expressing this molecule SPP1. But one thing that has not worked, but continues to really plague, I think, the myeloid world, is the signature of M1 and M2. And what I want to show you here is that if you take an M1 signature and N2 signature, it doesn't do anything to differentiate the populations. It may be a layer, but it's certainly not a layer that represents either maturation or any of these other things that we think are important. Some of the other things that do parse them very nicely, for example, is glycolysis. So we showed this, and this is what the basis for which I'll claim that all the macrophages that are called SPP1, which are the same as these, are in fact the same thing that have historically been called hypoxic macrophages. And they're very much focused on glycolysis, whereas OXPHOS is more spread across all the other populations. So if you take this data and you take a signature, for example, of M1-ness or M2-ness, and you just ask, are the genes within that signature cross-correlated in tumors? The answer is all this white. The red means that they're correlated with themselves. That's the diagonal. Genes, green genes correlated with the green genes, each individual one. But there's almost no evidence of any other structure in here. There's occasional splotch of the little red co-association. And the Pearson R for this just looks like it's neutral. Like there's really no association between M1 genes and M1 genes, nor an anti-association of M2 genes with M1 genes. So this is something that, for example, Dana Pear showed in, I think, 2021 in a setting, and yet it still dominates. Another way to look at this is this is a study we did in wound healing, where we took we're showing two of the most dominant genes that people associate with when they say, oh, I have M2 macrophages. There's arginase and there's CD206. And if you look across the differentiation trajectory of myeloid cells in wounds, some of these not even being present at the same time as others, so they're definitely individual populations. You can find this rise in arginase in sort of early differentiation in wounds, whereas this separate phase of expression is 206. If you saw these two things, you would not say this is a module. These are two uncorrelated sets of genes. And yet, immunology seems to be a bit trapped in this kind of all-inclusive thinking about this conception of M1 and M2. And I think it's just a matter of convenience for people. If they find these genes, they then find they can publish a paper referring to immunosuppressive macrophages or M2 macrophages. And to kind of give you a reason why I think it's important to get out of this is that despite all the TISNEs and the computational maps that we and others have done to suggest that it's more complicated, in the last 18 months, there have been over 12,000 papers, this is from Google Scholar, published that refer to M2-like macrophages. And they largely use CD206 as their basis for calling that, and they largely then refer to them as being immunosuppressive. So there's a series of correlations that are made that none of them are actually very good, and I think it's time to move on from that. And one of the things we decided to do was to say, okay, if people aren't watching the computational data and aren't sort of paying attention to the fact that these genes don't really mean suppressive or even a module, we said, well, what would happen if we just got rid of all the, quote, M2 macrophages, the ones that express this molecule CD206? And so to do that, we put into the mouse locus a cassette by which we could, into the CD206 locus, by which we could use an additional gene to provide myeloid specificity for all myeloid cells that express CD206. So essentially, by expressing, for example, CSF1 Cre, as they will mostly show you, we could end up with a locus in which any cell that wants to express CD206 will both become venous positive, so it'll be colored, and it will also have a death gene that we can access and turn and kill those cells. So just to give you a sense of this, the expression of these in macrophages is about a log higher than any other immune cell. Neutrophils and monocytes have very small numbers, and they're very, very weak also for the MFI of this reporter. There's a population of cells that you can call CDC2s. I think these are a population that are monocyte-derived CDC2s. I think Pierre Gremont-Pré and a few others would like to call these CDC3s, but that name has obviously been taken by about 7 or 8 other cell types by now, or cell populations. But in any case, we have very, very high expression in the macrophage compartment, both of this in frequency and MFI. So this allows us to do the following experiment, where we can take that mouse that I've just shown you, where the CD206 is driving venous and DTR in myeloid cells. And then we can give them tumors, and at various points, give them, for example, T1 cells, T cells that will reject the tumor. And also at various times, we can use diphtheria toxin as the death gene to hit the diphtheria toxin receptor and kill off those particular cells. And what's shown here is that there's 2 different patterns we're gonna use. One in which we can deplete very early, and one that we can deplete very late to understand what's happening. So I'll start with the late. So if we start late in tumor development, so right around day 10 in mice, and we're going to harvest at day 14, and we start to deplete with the DTX, we see this very nice depletion of tumor-associated macrophages. The CD206s go down from being almost 30% of the tumor to being very little. We don't have any effect if we do that on CDC1s, NK cells, very, very modest, if anything, and the T cells that we put in, very modest, if anything. So this provides evidence that this is a very specific way to get rid of tumor-associated macrophages, far more so than, for example, than clodronate, and it's also specific to the 206 population, which I'm not showing you here. But if we do it early, we, of course, still see the reduction of the TAMs, but we see then an indirect loss. We know it's indirect because it didn't happen just based on the DT alone, of CDC1 recruitment, as well as the recruitment of NK cells and OT1. So these are circled because these are the module I have just been describing as being reactive. This is NK cells, T cells, and CDC1s, which are this collection, this archetype of cells that can help you reject your tumors. And all three of them are down. So very much despite the paradigm of the CD206 being really immunosuppressive, in fact, they seem essential. And they seem essential in this early window of tumor development in order to get an active immune system there at all. You can show that this is really important because, for example, if you do that same depletion, when you put in T cells that in red will start to control the tumor, if you do that in the context of this DTR, where you've prevented the CDC1s and the NKs from getting in there, now the T cells really don't control the tumor anymore. So this gives you, I think, a pretty good sense that the role of these cells is pretty formative for setting up the conditions of reactive immunity. Why that is? So a big finding in that respect is that when we do that diphtheria toxin depletion of 206 macrophages, we lose CXCL9 expression. And that's one of the predominant ways that T cells are brought in to these niches, the reactive niches. And if we look, in fact, even though people want to call CD206 macrophages as immunosuppressive, you see this diagonal going through here of CXCL9 versus CD206. So if you're a CD206 positive, you actually also are CXCL9 positive. So really calling these guys immunosuppressive is missing the fact that they can be your prominent source of T cells. You can look at this in humans to get a further sort of reality check. So here we've made a signature that represents all the ones that are not CD206 positive and another signature that represents the ones that are CD206 positive. And then we can query various different data sets. And for example, you find if you look at the number of CDC1s, what we call a stimulatory dendritic cell score, that very much scales with the number of 206 in patients as well. In other words, what this relationship we're seeing between the 206, their CXL9 expression, and the presence of CDC1s is a very positive correlation. Whereas, for example, if you just look at macrophages of the other populations, macrophages alone, there is no relationship evident. And if we then, for example, look at the probability of survival across various different tumor indications here, you can see that if you're at ratio high, you do much better than people that are ratio low. So in other words, as you get to this far right, you become more likely to survive, which is obviously a good thing. So I want to summarize here and leave a little time for questions, but I wanted the summary to kind of focus on this idea that we're getting this expanding understanding and or path to reactive archetypes. And we're doing that in some ways harnessing the components of the dominant archetype, or figuring out how those are used and what parts of them we want to use. And this is really important because remember, if you talk about this emergent behavior, thinking that the immune system and the molecules of the immune system, cells of the immune system have potential to get together in the components you want. And so really the issue is what parts do you want to save and which parts do you want to wipe out? So I think it's fair to say you don't want to get rid of macrophages. They serve some key functions. You like CDC1s, but it turns out you need macrophages to get CDC1s. So the emerging story that I just told you is that these 206 positive tumor-associated macrophages have been seen in some ways as an opposition to this reactive module, the NK cells, the CD8s, the CDC1s, the sort of method by which they feed back that we've shown now quite a lot over the last 15 years. But we're now seeing that the tumor-associated macrophages are actually a key component that lets this biology happen. And what that implies is that a component of the macrophage needs to be considered as being a component of this sort of reactive archetype that you want to bring forward. And I won't be able to show it today, but we actually have a series of drugs now that can enhance this macrophage biology and indeed actually enhance this outcome. And again, it's by starting from the standpoint of what do you have, what do you want, that you can see that. And it's also going to be how we're going to plant that drug into the correct indication space. So I'm going to end here again, as I started with a movie. And in this movie, what I want you to get from this is the fact that a tumor microenvironment is, in fact, a place that has titrated concentrations of immune cells in various different interactomes. So what you're looking at is into a breast cancer model in mice. This is a polyoma middle T. And the black space here is a tumor. The green and the yellow and the orange are all various forms of what we used to think of as inhibitory tumor-associated macrophages. I think we're beginning to rethink exactly how to call those. And blues are T cells. And as this movie plays, you can see this kind of swarm behavior of T cells in this region here at 12 o'clock on this tumor. And that is really what is dominant. That's the interactions that are associated with poor immunity, but are in tumor growth. These are growing tumors. And this is the immune system that is essentially licensing the tumor to grow. And here in the top right, we have these collections of red dendritic cells as a kind of the reactive archetype. And you can see the blue color has changed on the T cells. That's because they're actually fluxing ions that change the colored behavior spectrum of the fluorophores in there. And you can also find macrophages nearby. And I think this represents the concept of a collective. Again, this goes back to almost 10 years now. But to really think about this idea that tumor microenvironments can be really spatially parsed into some of this key biology, in addition to being questions of overall density. So I want to end on a note, since I have 100 of you on the line, to introduce something that I think is very important. That is to make our science better, to do better science, but actually as a method of us to do that. And that's this 501c3 that was formed in the last 2 years. That's called Solving for Science, or Solving For. That's been trying to figure out how, as bigger communities like the one that's formed here in the Myeloid Network, how we can get people together across institutions to do things that are really useful. And again, I very much applaud the Myeloid Network for having done that and thinking about how do you get people talking across institutions. So collaboration is something that we're very keen on. But the one I really want to talk to you about is publishing, or really how we as a group curate our scientific understanding. How do we curate science? How do we know what's good? Well, we do that right now largely via this very imperfect process called publishing. And I just want to point out that there is right now a pilot study going on. And one of the brilliant things about bioRxiv, if you use it at all, is that you can place things there and they will go down their track, but you can get a priority date for when you first discovered something and put it there, and then it will go through this very imperfect system and come out the back end. But it also offered us the ability to do something we do in the lab all the time, which is, we call them A to B comparisons. If you get a new antibody in and you want to compare it to the old antibody, that's a good thing to do before you start using it for big experiments. We said, well, why can't we just use bioRxiv to do the A to B comparison? We can see how things work in the old system versus a new system that would be more formative for authors, faster, cheaper, more fun for reviewers, more inclusive of reviewers, reward reviewers for their work. And it's something we call Discovery Stack. And again, I won't give too much detail, but I'll point you to the webpage. You can read about it. But it essentially offers us the ability to help, in some ways, improve each other's work rather than knocking it down and trying to block it from being publication, which I think is what we all see one another doing. And the idea is that somewhere along the lines, if we can do an A to B comparison where we get our papers ranked by their our perceived quality, we can look at how the journals do and do what's called an alluvial plot and just say, well, does this system work pretty well, pretty much as similar to this one, and do a little bit of analysis of the data, both from things like author satisfaction, reviewer satisfaction, and overall how we think it curates the value of a piece of information. Important, obviously, in the world at large beyond science. So this QR code, if you have a phone handy and want to grab a screen snapshot, will point you to where this experiment is taking place. We need something we call peer improvers. That is, people that look through papers. And I'll say this is a lot more fun than peer review, where you have to write a report to a journal editor. It's a lot more formative, I think, so the authors can see what part of their manuscripts are failing. They don't have to read the tea leaves of the author of the thing comes back from the journal. We really encourage co-reviewers. I think that's don't currently in peer review really bring trainees into this process to say, this is how you should be doing in the future. This is how we do it now, to teach, in fact, this process. And they're also very much looking for papers in immunology, cancer immunology. It should be a preprint so that it can be going, and the experiment is just kicked off. This is being run by Maureen McGargle out of Solving For. She was a faculty at St. Jude's until recently. So if you want to be a reviewer, improver, or an author and want to participate in a study that can improve, make science better, please check this out. Okay, so I need to thank a lot of people. My work, our lab's work is really quite a big collaboration. I mentioned the archetype work of one of the bigger players in that has been Alexi Combs, who's now faculty at UCSF. Vincent Chan, through the ImmunoX initiative, even before it existed, oversaw a whole lot of lab members and members of his, kind of essentially became his lab and then Alexi's lab, that do a lot of the patient sampling. Ken Hu and Nick Kuhn did the wound healing studies that I alluded to. Tristan did the human murine. Arjo did the CD206. And there's a lot of other cool science going on in the lab of people that are pretty awesome. I'm pretty lucky. This is from a recent lab retreat. A lot of the people, major funding for this work is government funding. So they thank them as well. I thank the people. Okay, thanks very much. I don't know if there's questions. I don't know how this works, but I will look. There's a chat.
Thanks, Max. Yeah, we can start with questions. Thank you so much for the beautiful seminar. And I think my lab, at least, and I think a lot of other people on the call have taken the oath to never say M1 or M2 again, but that computational proof is from you and Dana Pierre that you had alluded to is so powerful, right? That these states don't exist in nature and biology. So thank you for that, as in addition to the beautiful work you showed. We can start with people raising their hands and turning on their cameras to ask questions, and then we can go to the chat. Jelani, I think you had your hand up first.
Hi, Max. Great talk. We work on CD206, and we came to a similar conclusion about the mannose receptor. And so we found that if you alter the metabolic states, macrophages still express 206, but they became more inflammatory, had a Th1 response. And we found that this was through a process called FGAR that regulates nucleotide synthesis. So I was wondering if you all had looked at the function of these cells that maintain the mannose receptor expression and what they made, what they did, et cetera.
Really fascinating. No, we haven't. And in fact, we find ourselves buried in a plethora of riches with these archetypes where the relationships between one thing and another, and also even within cell types, the relationship between one thing and another is just almost more questions. Well, it's more questions than we can answer. But I'd be delighted to talk to you about that more. And I know actually Arjo, who's coming to Johns Hopkins as a faculty, you two should definitely get together. He's a great guy. You enjoy working with him, I'm sure.
Chris Garris.
Okay. Oh, fantastic talk, Max. I had a question about the CD206 macrophages and the degree of vessel normalization that occurs within these types of tumors, and whether sort of these macrophages could be laying the vascular network for efficient T cell migration.
Well, I think we haven't looked at the vascular network like visually so much in these knockouts. We've used the Venus to characterize where 206s are, and they're largely, like in an MC38 tumor, for example, which is like one big ball, they're largely on the outside more than they're on the inside. But macrophages in general are a bit too. I think there's something to what you're alluding to. I don't know that we have any data ourselves to support it, but the idea that you put your recruiting cells like your CXCL9 positives and even the CDC1s, I think in lymph nodes, there's pretty good evidence that CDC1s and actually no, Torsten Meppel has seen a bit of it also in tumors, that the CDC1s tend to be closest to the vessel. So this idea of maybe direct recruitment and having therefore the base of operations for those foci to be there, I think is reasonable. It's a reasonable summation. In terms of vessel normalization per se, though, that we haven't looked at that. It's an interesting question. Thank you.
Santiago, are you on? Do you want to ask your question, or I can read it? So Santiago asked, when looking at the archetypes versus Ki67 expression, it seemed that apart from the immune desert clusters, the DC1 clusters were also fairly high for Ki67. Do you reckon this could indicate in situ DC proliferation in these tumors?
Yeah, so the interesting thing about Ki67, I'm going to go down a brief rabbit hole. All tumors divide. If they didn't divide, we wouldn't care. What the Ki67 protein positive refers to, I think, is how they divide. And if you read up on Ki67 a bit, or if you know it, it's a cell cycle checkpoint, not an immune checkpoint, a cell cycle checkpoint. So the cells turn it on after S phase to allow themselves to go through cytokinesis. And then they're supposed to degrade it. So when we see Ki67 high, one of the suppositions, and we haven't gotten deep into this, but one of the suppositions is that maybe in those tumors, the way that they've started to divide and to get themselves going is to specifically mutate things associated with degradation of Ki67. And then your observation is our observation too, that it's not just the tumors that are doing that, but it's also immune populations actually show higher Ki67. Everybody in those tumors show higher Ki67. And so if you take that logic, it's possible that what those tumors, the immune deserts, represent are ones that are perhaps, for example, protease deficient, or where the tumor has figured out a way to express something that essentially changes the protease background by which Ki67, the protein, is normally degraded. And I said that in part too, because when we look at Ki67 gene expression in cross tumor types, it's only modestly higher in those deserts. It seems like the protein expression, not the RNA, which is how Ki67 is normally, is the thing that's dysregulated in those tumors. Other tumors presumably get through cell cycle in other ways, maybe just even conventionally using growth factor pathways and overusing oncogenesis. But your observation is exactly something we've seen too, that a lot of the population, not just the tumor, are Ki67 high. And again, that points to something that might be very fundamental about those tumors and their uses, for example, of something like a protease, which then proteases get you very quickly, if you're an immunologist, myeloid biologist, to think about antigen presentation and maybe even vascular formation. So there's definitely something to investigate there, and really kudos for picking that up.
Yeah. Sneha Ramakashrina, you have a question that sort of overlaps with a couple people. Do you want to ask that?
Yeah, I think, thanks so much. This talk was awesome. My question is really sort of thinking about what the actual drivers are of these various immune archetypes, and specifically, I think these are all markers that we use, but what are the things that truly drive the state of these myeloid cells, and can we target those to then skew in the direction of immune activating, for example?
Yeah. Well, again, one of the things that we're seeing that's pretty interesting is that when we look at the mutational landscape of the tumor, we're also finding that certain mutations in certain oncogenes and other genes are parsing into archetypal space. Push that up against the fact that there is a tissue-specific bias towards an archetype. We saw kidneys on one side, there's a lot of the melanomas. And I think what's basically happening is that the tissue has an inborn advantage in harnessing certain immune archetypes. Certain things are often seen in the tumor, like in fact, viral infections. So certain things are already seen there. And so the immune system is already in some ways poised. And then the tumor, in order to get that state to be locked in that it can grow with, doesn't have as much work to do, and maybe has specific oncogenes that it can turn on, or various genes that it can mutate, that essentially makes it look like the thing that the immune system looks at and says, oh, this is a wound in need of final repair, or this looks like adipose tissue that's in need of essentially metabolic tuning, or what have you. And so our conception of that, of the answer to your question, is that there's all these biases that bias towards how a tumor will quote unquote win, and how it wins is to get into an energy state, or like a gene expression state, where it's expressing a sufficiently similar signature that all of the immune cells see what they expect to see in that setting. And so I don't have, basically what I just said was, I don't know what those genes are, but I think the conception that we have here is that there are collections of genes, and like I say, with this mutational analysis, we're starting to see some of them that parse into the compartment, but which ones actually then talk to myeloids directly to affect their density or their gene expression, we don't yet know. So, but it's also a very good question. Thank you.
Helen McRae, you had a similar question. Do you want to turn your camera on? I think there's a part of it that wasn't answered yet.
Hi, yeah, thanks for a really interesting talk. Yeah, I guess I just was wondering if you could comment a bit more about kind of the distinction between CD206 expression and the whole macrophage population being there. Like, is it CD206 itself is doing something to promote that immune response, or is it more just, has anyone tried those experiments? I'm not sure if your lab has or someone else, kind of just reducing the expression of CD206 as opposed to getting rid of the whole population?
Yeah. There is a CD206, I think, a knockout. I don't recall it having a particularly profound phenotype, but I might imagine Jelani would know that more than I would. I assume that it leads to various forms of, because of the scavenging function of 206. But I don't recall it having like an all or none response like this, where all of a sudden, you can't get recruitment. So I think the association I'm showing, and I think we should all be wary of association, but the association between CD206 and CXCL9, I think, is what we think is probably the most important. CXL10 is also pretty well associated, is what we think is most important for what we're seeing, namely that function of cells that often express TO6 is that they also often express the major chemokine for recruiting kind of reactive immunity. Whether you can, and one of the issues I think about modular gene expression, when you think about collections of genes, and maybe 206 and CXL9 are part of a frequently like a super enhancer kind of situation, is whether they're obligately tied, and whether you can dissociate that. And, for example, get CXL9's 10 without some of the things that you might not want to. Again, macrophages aren't always, I'm not saying that the 206 positive macrophages are always good. I think later in tumor development, they definitely have a role in talking to T cells and overstimulating them to exhaustion, making them less effective killers. So, dissociating this concept of individual genes, modules of genes, and then also ultimately function, I think, is still to be determined.
Thank you.
Joanna, do you want to ask your question, or I'm happy to read it if don't come on?
Yeah, I can ask it. Thanks a lot, Max. That was such a really thoughtful talk in many respects, including what you shared at the end. Obviously, I think many of us agree that the publishing system has a lot that needs to be fixed. My question was, it kind of relates to one of your responses to a previous question, basically about how these archetypes come to be, right? And the contribution in terms of the spatial architecture, the potential role of the extracellular matrix in defining, in modeling, in remodeling those archetypes, particularly after therapeutic interventions. So I'm interested to hear your thoughts on that.
Yeah, well, there's a few. Like if we take mouse models just as an example, and I'm referencing this work, really nice work from Christine Lucian at Genentech. Mm-hmm. When we look at a model like B16, eventually the ones that grow out all seem to have very similar composition. And again, if we parse out mouse models, the B16s can be very different, for example, than Renka. And so what I kind of take away from that is that as a tumor is growing, eventually the tumor itself can be very informative as to what the immune microenvironment looks like. But if you look at the earlier part of Christine's work, like the very early time points, it's quite a lot more dramatic. She does this thing where she puts a little bit of tumor into all these various different holes in the back of skin of ears, and she'll see some of them that are filled up with T cells, and some that are not, and some that are rejected, and some that are not. So it looks to as if in the early phases a tumor maybe doesn't have enough oomph of its cells to basically dictate everything. So that's one thing, to your point of I think the tumor itself has a lot to say. It's gene expression about what sorts of immune archetype you get, which is to say then that when we look in the humans where we actually have paired primary and Mets, they often, I'd say I think we only have 10 of those, but 9 out of 10, I think they're the same. Like the compositional, what's high and what's low is the same. Mm-hmm. But the 10th is not, right? So I think all things are probably possible in biology. But there is a predisposition that if you have and this can also go towards the idea of metastatic seeding, where you seed, that there are obviously receptive environments that look more like the tissue you came from. And so you are better tuned, in gene expression to set up that same tumor microenvironment, those settings. But that 10th can imply that, one of the things that tumors can do is they can mutate. And even if these archetypes are quite different in gene expression, but really a tumor just has to win. It just has to figure out, the right combination of site and the genes that it can mutate to maybe make it somewhere else. With regard to the extracellular matrix, the roles, I mean, I guess I would take that very naive standpoint that, metastatic seeding as an example. After therapy, I actually had no data, but after seeding, that, some of the tissues that have similar either ECM laid down, or maybe even some of the same sort of pattern, wince, frizzled, notch, et cetera, in those tissues would be the most receptive for that tissue to seed there. And what I've just did is I've just completely, almost like a politician, I've completely avoided your primary question, which was like
I was waiting for you to get to it. Very Kamala Harris like.
I know. I was reading the New York Times about her too. I didn't realize it's a thing, but I ultimately think those things do wrap around your question, but I don't actually have any data. And it's really harder for us all to get data where if you wanted to get only a red archetype of kidney cancer, so you're still 40%. So you got to, like, the amount of work to do to see that in patients is very tricky to get and obviously patient consent. And I don't know if you have it better than we do, but to be able to even get to tumor tissue multiple times from the same setting is really requires quite a lot of Herculean IRB and
maybe I can just quickly add a quick follow-up question. Are you looking at all then? I mean, of course, the organoids and bioengineering is very hot at the moment, and that would be in principle a way to design and to basically create certain archetypes. Is that something that you see any future in doing?
Yeah, well, I mean, we have a study where we're essentially trying to ask, between two mouse models, one's very myeloid rich and one's very myeloid poor. We're trying to ask if we can identify the genes that are driving that. So I think there are things to be done with model systems. We just have to be careful we model something that's real. It's actually the method that happens in situ. But yeah, I think that's a good way to go.
Okay, thanks.
Thank you.
Okay, well, thanks, Max. I think that most of the questions have been answered in the chat. Certainly, I think if others have more questions for follow-up, they can shoot you an email. Thank you so much. And thank you to everyone else for joining the call today. Have a great November. Hope to see you at SITC, and then we'll see you in December back for the Myeloid Network seminar series.
Bye. Thank you.
Bye, Joanna. Thanks, Max.